Ladies and gentlemen, I'm Jacob Huang. I'm the Executive Director of IAS CDU, and welcome to this IAS Distinguished Lecture. You know, we do have a very small community for neuroscience in this university, but still we have a crowd today. So I hope maybe in 10 years, this room will be all packed. Yeah, so we're going to have a very huge neuroscience programs in the future. So what do you think? Yeah. Okay, that's all your duty or your responsibility to attract more people, younger or senior, okay? So today we are very, very honored to invite Professor Tomas Hökfelt.
Perfect.
Okay, and he's our senior fellow of IAS in CDU. Currently we have 26, and he's one of them. And also he's a professor at the Karolinska Institute, Sweden, and also member of Swedish Royal Academy of Science, and also member of Academia European. So also foreign members of National Academy of Science in USA. I got some more information from the website. I think he's also the member of International Scientific Program Committee for the 6th IBRO World Congress of Neuroscience. This is a very, very important person, scholar in neuroscience. And member and chairman for the committee are Dragat and Minena Foundation, yeah, sorry, yeah. And also, you know, he's the member of Nobel Assembly. That means he elected the Nobel winner, the laureate. So which one is the new one? Okay, yeah, okay. So whenever we have this workshop, lecture in November, everybody gets so nervous, yeah, because one of them might win the Nobel laureate. So CDU is not so far away from that. Yeah, probably not our local faculty, but our visiting scholar or a senior fellow, they do have chance. So very interesting. And also he's the member of advisory board for the Institute of Neuroscience of the Chinese Academy of Science in Shanghai. And I think for the time being, he is a professor in the Department of Neuroscience or at the Karolinska Institute in Stockholm, Sweden. And he's also the world leading in the mapping of chemical components in neurotransmission in the brain. And also the member of Scientific Advisory Board at Neuron Company. Right? Yeah. So he's not only the professor, probably also a big boss in the company. Yeah. And I think he pursued his undergrad, master, and PhD program all at the Karolinska Institute. In 1969, he was appointed as assistant professor in the Swedish Medical Research Council, and later in 1979. He becomes a professor in this university in histology with cells biology. Yeah, and in 1993 to 1999, he served as vice chairman of this department of neuroscience, and he received many awards, so many. Yeah. For example, like 2 awards from the Royal Academy of Science in Sweden, and a New York Academy of Science Award in Biology and Medical Science, and some other like Artois Lator Health Prize. What is so complicated? Okay, yeah, like you've got some Chinese like Sao Yi Fu, and the Western people say, what does Sao Yi Fu means, right? Yeah, so, and many other prices that I can hardly pronounce, sorry. And in 1991, the Eli Lilly Clinical Research Award and many others. And from the Google, I checked this morning. I got shocked. Do you know how many Google citation of Professor Hökfelt? Yeah, usually people say, oh, I got 10,000. Oh, okay. Oh, I got 20,000, and he got 120,000. In Chinese, 1, yeah, 120,000 times. Takes a lot of effort, right? Yeah. So, and also his H index is 172. Yeah. So that's really, I think many papers got a citation about 1,000 times. So if you got one, it's already impressive. So got many. So Professor Hökfelt is really the giant pioneer in the neuroscience. So let's give a warm applause to welcome Professor. Thank you.
No, I don't think that even if you told so much about me that most people here know about me. So I wanted to give you a little bit about my background, why I am here, who is behind me. And I am a histologist, and that means that I use a microscope to look at tissues like the brain. And I take a photo of something in the brain. And my whole career is built on photos that I have taken in the microscope, and they have started some new project. So perhaps the most important photos that I ever took were these two. And as you can see here, there are two photos which are pretty similar, and it's from the superior cervical ganglion. What is that? That is a part of the autonomic nervous system. You see here the sympathetic nervous system and the parasympathetic peripheral nervous system. And this particular pictures were from this ganglion up here, which supply the eye and the salivary glands. And what we observed was that there were two types of molecule in these cells. And I come back to exactly in a few seconds what that was. But when we looked through this peripheral nervous system, we saw that it always was like that they did not only have one, but they had two. And you could subdivide this nervous system on the basis of these compounds here. Now, what we see here on the left, in the left picture, is a peptide, which is called somatostatin. And this peptide was discovered by, unfortunately, the man who is not quite in the picture here, because with this projector here we have a little different. But it's Professor Roger G. Mang at the Salk Institute, and he discovered somatostatin, and for that was part of the reason for giving him the Nobel Prize in 1977, together with Andrew Shallow and Rosalind Yalow. So this was a hypothalamic hormone when it was discovered, which inhibits growth hormone release. And that's why it got the name somatostatin, inhibiting growth. And what was a surprise then was that this hypothalamic hormone also was found in the peripheral nervous system. So this was a surprise. On the right-hand side here, you see a staining for an enzyme called dopamine beta hydroxylase, which is the enzyme that converts dopamine to noradrenaline. And noradrenaline is, of course, the transmitter in the sympathetic nervous system. And the discoverer of noradrenaline was Ulf von Euler, a Swedish scientist who got the Nobel Prize for that discovery in 1970. So what this says is that these cells here, they contain both noradrenaline and somatostatin, this peptide. And that means there are two transmitters in the same neuron. I'll come back to that very soon. Now, we published that in 1977, so it's 40 years ago. And ever since then, this has been my main aim to understand what this means, that neurons have two transmitters. Now, why did I start to work with microscopy to look at the brain and the nervous system? This is because this man here, Hillarp, you have never heard his name. Most people have not heard his name because he was a very shy man, actually, and he came to Stockholm in 1962 from Lund in South Sweden, where he, together with his younger collaborator, had developed a new method which we call the Falk-Hillarp technique. And this was the first method which you could use to see in the microscope a transmitter molecule like noradrenaline. And he came in 1962, and within a few years he recruited 10 young students, and I was one of them. And as you see here, he actually only lived for 3 more years. So he died in 1965, so very sad and very tragic. But he was my mentor, and he started my project. And this is Arvid Carlsson, who is more well known because he got the Nobel Prize in 2000 because he discovered dopamine as transmitter. And when Hillarp died, none of his 10 students had completed the thesis, defended the thesis. And it was possible for me to go to Arvid Carlsson, who worked in Gothenburg in West Sweden, to get advice from him because Hillarp had died and we were, so to say, alone. Now, the first of Hillarp's students was Kjell Fuchse and Annika Dahlström. And here you see what we have on the board in our department, all theses from the first student of Hillarp to 2001. So these are the 10 theses from his original students, and none of these theses were defended before he died. So he died before he saw that any of these students completed the thesis. But the first thesis is a very famous work by Kjell Fuchse and Annika Dahlström, because here, for example, there were 4 papers included in that thesis, and the most famous one is this one here. It's a supplement in Acta Physiol Scand, which now has almost 5,000 citations. So you don't have to publish in Cell or Nature or Science to make something that is of lasting value. You can publish in a very simple journal, provided it's good stuff and important stuff. So what Kjell and Annika did was to define the catecholamine neurons in the rat brain and the 5-HT neurons. So they said the noradrenaline neurons have A1, A7, and then the dopamine neurons A8 to 12. Among the dopamine neurons are, of course, the substantia nigra dopamine neurons, which degenerate in Parkinson's disease. So this was a fantastic work, and it means this is what it looked like in the microscope. You know, this is locus coeruleus, which I will talk about later on. And this was the first time that one got to know that the locus coeruleus neurons, which are so important, are noradrenergic neurons. They have noradrenaline as their transmitter. Then I have one more person that I'm very grateful to, and that's Menek Goldstein. Menek was a professor at the New York University Medical School, and he purified the enzymes that are involved in the catecholamine synthesis, tyrosine hydroxylase, dopa-decarboxylase, dopamine beta hydroxylase, which was the one I showed before, and the last one, PNMT. And he made antibodies, and he gave us these antibodies so we could use a technique which is called immunohistochemistry. And that was the first pictures I showed you were immunohistochemical pictures. And this was a technique that Albert Kuhns, who was professor at Harvard in pathology, published already in 1942. So it's a very old technique, but from 1942 to 1969, only microbiologists used this technique to identify bacteria and so on. No one used it in neuroscience until this group in Australia, and we started to use them thanks to Menek here. And nowadays, of course, immunohistochemistry is very much used. I looked it up here. You see 2016 in PubMed, and you see 626,000 hits. And then I looked up a little later, and then I found 641,000, and then I calculated that there were 75 new hits every day. So it's one of the most used techniques because it's so general. Whenever you can make an antibody, you can also use immunohistochemistry to study the localization of this molecule in tissue section. So that's a very valuable. Menick, he died quite a number of years ago, and he was living alone, and they found him dead, sitting in his chair in front of his computer, and on the screen was an application for NIH grant. So he worked until the very end. Very sad. When I started my histochemical work, the nervous system was very simple. There was a rule which was called Dale's principle. Dale was a very famous scientist, saying that every neuron only releases one transmitter. So here the transmitter was released from nerve ending and then acting on receptors in the dendrite. What the work did that I showed you was that it kind of violated this rule, because we showed that these neurons have two transmitters. So we call it coexistence of a small molecule, noradrenaline, and a neuropeptide, which is a bigger molecule. Now, of course, today it's much more complicated. We know that one single nerve ending can have many different transmitters. And we know that the peptides are stored in a special type of vesicles, the so-called large dense core vesicles. The classic transmitters like GABA and glutamate and dopamine, they are stored in these small synaptic vesicles. And we also know that peptides are released extra synaptically, here on the side, so to say. And thanks to my most successful student, because he now has a Ferrari car, he became head of global research first at AstraZeneca and then at Eli Lilly in United States. So he made a fantastic career. But he was also a fantastic scientist, because what he found out was that the peptides are not released when neurons firing at normal activity here. Then the classic transmitters are released here. Only when neurons are starting to fire at high frequencies or burst firing, that's when the peptides are released and have their action. And that was a very important finding, and has several consequences. And as I said before, this has then been my goal in life, to understand what does it mean that a neuron has more than two transmitters. And what I will talk about later today is to answer the question, is this also important in the human brain, and is it also important perhaps in disease that we have more than one transmitter? Because we have to think about this. And for example, if you are analyzing peptide signaling, this only occurs when neurons are firing at a high frequency with high intensity. But then you also always release the classic transmitter that is stored in the same nerve endings, because they have already been released when the neurons fire at lower frequencies. And you could also say the other way around. If you analyze small molecule signaling, are sometimes peptides also released? That's something you have to think about, but then probably only when the neurons are firing at high frequencies. So things have got much more complicated than in the early days. So the family of transmitters that we have been interested in, and Professor here also, are the neuropeptides. And the man who really was the father of neuropeptide was a Dutch scientist called David de Wied. He coined the term neuropeptides. And his student, Peter Burbach, made this very in-depth analysis of how many peptides do we really have by looking at the genes. And it turned out that we have more than 100 peptides. So it's the most diverse in terms of number family of transmitters. And there is a correspondingly large number of receptors, which always are of the 7-transmembrane G protein coupled type. And we believe that they are potential targets for drug development, because we know that 40% of all drugs that we use today are working via 7-transmembrane G protein coupled receptors. So this is something we have in our mind, that maybe this can lead to development of new drugs. Now, with time, we understood that peptides are not used every day, because it's only when neurons firing at high. So this is a special situation when the nervous system is stressed, when it's damaged, nerve injury, perhaps with mental disease, perhaps with addiction and so on. So we have got a new perspective on what peptides do, because at one point people got more and more disappointed about peptides. So during the 70s and 80s, there was a hype. Oh, important peptides. I was invited every week to a meeting to give a talk. And then came the downhill period. And this was very nicely said in this paper here by a colleague called Chancey Bowers, because he had a title of superfluous neurotransmitters. And he meant the neuropeptides. So they have no function at all. So that was not good for me, you know, when I applied for grants, and you know, superfluous transmitter. So now this has been my next goal, to really show that this guy was wrong. So the peptide that we have done most work on is Galanin. Just like Professor Heer's work on CCK, we have been working on Galanin. And Galanin was discovered at Karolinska Institute by these two scientists, Professor Victor Mutt and his PhD student, which was a Japanese PhD student. And here you see the size. It's a 29 amino acid peptide, so it's a bit bigger than CCK, and it's very conserved here at the N-terminal end, and then there's variation here. But it's not a very popular peptide compared to other peptides. For example, neuropeptide Y, which also was discovered by these two, has been, if you look in the PubMed, you get 14,000 hits versus just 4,000 for Galanin. And orexin, which is one of the most important peptides nowadays, has already 4,000 hits, even if it was discovered 15 years later than Galanin. So Galanin, we have been not so many groups working on it. So we have been a little bit alone. I know that Professor Heer also has worked on it, and that's what I like very much, and I hope that you continue to work on it. So nevertheless, what one has to know is that Galanin acts via 3 receptors, and GALR1, GALR3, and GALR2. And as you can see here, GALR1 and R3 have a similar mechanism. They cause hyperpolarization by opening potassium channels. So that, please remember that. Why did we got interested in Galanin? Because Galanin was discovered pretty late, 83, as you saw. Well, we did some early experiments looking at dorsal root ganglia. And these are, here you see the cell bodies in the ganglia, peripheral branch to the skin, central branch to the spinal cord. And among these are the pain neurons. And what we observed early on was that if we looked at the normal ganglion, then there were only very few cell bodies. But if we did a lesion, if we cut the sciatic nerve, then suddenly there was a fantastic upgrade, 100-fold increase in synthesis, and that we thought was very interesting. So these were also these pictures that really changed my mind, you know, that really made me go for this peptide. And we have studied a lot of these things, this peptide, for involvement in pain and regeneration, but I'm not talking about, I'm just telling you this because this is the first finding that really made me say we want to do research from now on Galanin. And the next picture is another one of these very important pictures for me, and that was the locus ceruleus that I told you has noradrenaline, shown by Kjell Fuchs and Anneke Dahlström already in 1964. Because if you look here at the locus ceruleus, you see this is a very nice little compact nucleus in the pontine part of the brainstem, and you see that tyrosine hydroxylase, every neuron has tyrosine hydroxylase, the first enzyme in the catecholamine synthesis. And as you see here, many of these cells have Galanin also. This was my student, Tor Melander, who made this discovery, and he also saw that the serotonergic RAF neurons also have Galanin, and there are also some other neurons here. So this made also increased our interest in Galanin, and especially since these systems, both are the 5-HT and the serotonin neurons, are interested in stress, anxiety, and mood disorders. So the rest of my talk will now be on depression, and I just tell you I'm not a psychiatrist. Even if I talk about depression, I actually spent 2 months in a ward when I studied medicine, but that's many years ago. So what I'm saying now is not a specialist in psychiatry, but I talk about depression in any case. So we know that depression is a very serious disease. 5% of people will be affected by this disease, more women than men. And what is very worrying is that especially young people are affected by depression, and one serious development could be, of course, suicide. So there are treatments, pharmacological treatments, mostly is like of these reuptake inhibitors like Prozac, SSRIs, but they have side effects, and about 1/3 of the patients are resistant to this treatment. There is something new, and I'm not going into that. It's a new drug called ketamine, which has changed the situation a bit, and we will hear more about that, I'm sure, in the future. But there has been a search for new antidepressants, and what I want to tell you is that maybe some drug that acts on the Galanin system may be a potential antidepressant. Now, the first peptide that was associated with depression, and actually in the clinic, was substance P, which was discovered by von Euler, also the same who discovered noradrenaline and got the Nobel Prize. But I want to talk mainly about Galanin here. But first I tell you about this first study on substance P, which was published in Science in 1998. And this was a fantastic study for anyone working in the peptide field, because what this study showed was that a substance P antagonist is as efficient as Prozac in treating depressed patients. And this came as a this was Merck, the drug company, and they thought they were going to take the whole market for treatment of depression. And the problem was that when they then expanded this study, as you will see next slide, this didn't materialize. The fantastic thing with this substance P antagonist was that there were no side effects here. This is the paroxetine, the Prozac-like drug, and here no side effects, just like placebo. But as I said, when they went to this big phase 3 trial, it failed. So Merck fired thousands of people because they had so great hopes for this drug. And it was also very bad for the peptide field, because after that hardly any company dared to go on with peptide antagonists for treatment of various diseases. It was kind of a real no. In any case, what we decided to do was to work on Galanin then. So we formed a Galanin team for depression, and Vincent Pierrebon and my Chinese collaborator especially worked very much on this, and other people also. So we have been working on that now since many years, and also many other groups have been working on it, and this is a summary of the work on animal experiments. We have depression models for in rat models for depression, and I just say what the conclusion was from all these studies was that the Galanin R1 receptor is pro-depressive. So if you want to treat these animals, you want to have an R1 antagonist. But also GALR2 was interesting because it's antidepressive. So here you would like to have an agonist to treat depression. So what I then was thinking about was, are these animal experiments really relevant for human disease, for human depression? Because we work on mice and rats, and we think that this is relevant for humans, and drug companies work on mice, and they start to develop drugs on the basis of results from animal experiments. So I decided that we should really look at the human brain to see if it's the same receptors and so on. It's a big step to go to the human brain, of course, because just the number of neurons is so 1,000 more neurons in the human brain than in mouse brain and so on. So and it's a very difficult thing to work with human brains, because you have to work with post-mortem tissue, and you have the post-mortem times that vary, and so on and so on and so on. But we decided to go for it in any case, and I had a postdoc, Ervan Lemaitre, and then other people have also joined. This is a Hungarian colleague who has a brain bank, so we got the tissue from him. And we focus here now today on locus coeruleus. And locus coeruleus is here a little nucleus also in the human brain, just like in the rat. You can see it by eye because it has a pigment, and that's why the coeruleus means blue, and so you can see it by eye here. This is a little nucleus that we have been looking on, and this nucleus is very special because it is just a small number of cells, and they project all over the brain, to all parts of the brain and to the spinal cord. So it's kind of a controller of the entire central nervous system. And again, we got some pictures that were surprising. Because, and that was Ervan who showed me these auto in situ, and this is now a new technique. It's not immunohistochemistry any longer. It's in situ hybridization, and that shows the mRNA, the transcripts for the enzymes. So what Ervan showed to me was that when he put on probes for tyrosine hydroxylase and Galanin and for receptors, he could see that, of course, as expected, the cells have tyrosine hydroxylase. They are noradrenaline cells. And they have Galanin, and that was exactly as in rat. But the surprise was that it was not like in rat, the R1 receptor. It was the R3 receptor. And of course, if you want to develop drugs, you cannot develop against the wrong receptor, so to say. And as I said before, we had thought it would be good to have a GALR1 antagonist, and now we see it's GALR3 that is important. What you can see here, if you want to detail, is that you see that here the enzyme is in all cells. Galanin is not in every cell, And some are very strong and some are very much weaker. The receptor is strong like tyrosine in all cells. So that was an important finding. So there are species differences, and that was in principle very important to show this. And I have summarized something here, and I just show the important here. For example, if you look at locus coeruleus in blue here, rat, mouse, and human have galanin itself. But if you look at the dorsal raphe, the serotonin neurons, only rat has galanin. And then if you look at the receptor, the R3 receptor is in human, but not in rat and probably not in mouse. So these are clear species differences, very important for development of drugs. Now, then we did a very big experiment. We got brains from a Canadian Montreal brain bank, Naguib Meshavar, here. And what they did, they dissected 5 brain regions. Locus coeruleus, just the one I've talked about all the time. Dorsal raphe, this is the region where the serotonin neurons are. And 2 regions in the prefrontal cortex. And this is work that this my graduate student Swapnali Bard has done. And she did 3 things. She looked at the mRNA levels with qPCR. She did pyrosequences to look at methylation, that means epigenetic analysis. And another group also helped to do the measurements of galanin levels with radioimmunoassay. And this was done in both male and female brains, and both in normal control brains and in brains from persons that had committed suicide. So we had more than 200 samples that Swapnali analyzed. And we got very much information from that, interesting. We got information from the control values that we didn't know about. For example, what are the levels of transcripts for galanin and the galanin receptors? What are the difference between male and female brains and so on? And I just show you few from the normal brain here. Shows, for example, that if we look at female controls, the levels in the locus coeruleus are 25 times higher than in cortex. And that's in a way expected, because this is where the cell bodies are, and this is where the terminals are. We also can see, for example, of the transcript levels in the controls. Now this is then raw values. So if the numbers are low, it's higher than the numbers are high. So this means that if you compare the same, the cortex here and the locus coeruleus, then it's about 6 times more in the locus coeruleus than in cortex. So these are control values. And let's jump. What was also very interesting was that in the controls, if we looked at the 2 cortical regions, which are pretty close to each other, the levels of the GALAR1 receptor was 8 times higher in the dorsolateral prefrontal cortex than in the anterior cingulate cortex. So there are very strong regional variations which we had no idea about before. But then comes, of course, the interesting and important comparison, that is between depressed suicides and controls. So what are the changes? Are there any changes in the depressed brains? And that was quite interesting, and there were dramatic things. I am going to jump to the next one here, and this just shows the raw value, so to say. But the next slide, I will show you the overview, because these changes were very selective. If you look here at this one here, you see in the anterior cingulate cortex, there are no changes at all, completely blank. If you look at the GALAR2 receptor, nothing at all. So the changes are up here in the prefrontal cortex, or here in the GALAR3 receptor mRNA. So this selectivity was very impressive, I think. We made us very excited about this. And what was more important was that when we looked at the epigenetic changes, so you see here we have 3 sites for epigenetic changes, and they are going in the opposite direction. If mRNA goes down, the epigenetic changes go up. The same here, goes up here, goes down here. And that's exactly what you expect, what you should expect, because we are known that methylation is inhibiting translation. And what I then want to concentrate on is the locus coeruleus. What you can see here is that we have upregulation of Galanin transcript in locus coeruleus and also up here in the dorsal raphe region, and also upregulation of the Galanin 3 receptor. So this means that the increase here is both in ligand, in the transmitter, as well as in the levels of the receptor. And this is then the basis for our idea of how perhaps depression can develop. These are the data that I build our hypothesis on. And then we focus here on the locus coeruleus system, neurons in the lower brainstem projecting to cortical regions. Here is the locus coeruleus neuron that we see here. And we consider the situation when the neurons are firing at a high rate, because we believe that the increase in Galanin in the locus coeruleus is a consequence of high firing of these neurons. These neurons have been activated by stress initially. We concentrate now only here on the cell bodies in the locus coeruleus. Of course, many things happen here in the forebrain also, but I only want to concentrate here on what happens in the locus coeruleus for this hypothesis on the role of the locus coeruleus and Galanin in depression. So here I have outlined what could happen here based on the data that we have obtained from post-mortem brains from patients that have been suffering from depression, from depression and committed suicide. So the first thing that happens is that stress induces an increased firing of the locus coeruleus neurons. And that's the same in rats, we know that. And that in turn increases release of Galanin. And since this is a peptide, the loss of peptide by the release has to be substituted by an increased synthesis, that is increased levels of transcript in the cell bodies in the locus coeruleus. So we have release of Galanin, upregulation of Galanin synthesis, increased mRNA levels in the locus coeruleus. That's exactly what we have seen. And also, surprisingly, which we don't understand, but also Galanin R3 is upregulated. Now, you may remember that I told you that GALAR3 receptor is inhibitory. It inhibits the neurons, inhibits the firing when that is activated. And what we believe is that the Galanin is not only released from the nerve endings in the forebrain, but also from the soma and the dendrites of the neurons. We have some evidence from rats, but it's also well known and very convincingly shown in other peptidogic systems, the hypothalamus, for example, that peptides are not only released from the nerve endings, which is the classical idea, but also from the cell bodies and the dendrites. So what we believe is that in this situation, we have higher levels of the Galanin 3 receptors, we have higher levels of galanin in the cell bodies, we have higher release of galanin from the dendrites, activating this increased number of galanin 3 receptors. And we believe that this is kind of a initially a protective mechanism to prevent the neurons from go crazy, to fire too much. So this should keep down the activity of the locus ceruleus to an acceptable level. So we think this could be a part of a mechanism that we call resilience, because we know that many patients, many human beings, they can tolerate a lot of stress. And one is always surprised, so why do they tolerate so much stress, whereas other people actually get depressed from too much stress. And this is called resilience, the ability of the organism to resist against stress. But at one point, this mechanism is overtaken. It cannot any longer work. And if this inhibition of the noradrenergic neurons is lasting for too long time, is too strong, then eventually the noradrenaline release in the forebrain, where actually the depression is formed, so to say. You know, I mean, depression is not a thing of the brainstem. It's in the hippocampus, in the cortex, and so on. So then the levels go down, and that is then the start of a depressive disease disorder. Now, if now the GALR3 receptor is so important here, the question is, of course, would a GALR3 antagonist be a good antidepressant? And the problem in the Galanin field has been that, contrary to substance P and PY and all these other peptides, many antagonists have been developed. And the only antagonist in the Galanin field that is a small molecule that passes the blood-brain barrier is a GALR3 antagonist. And that was developed in a small company in the United States. And by this man was the project leader there. And of course, this was already 2005. At that time, we didn't know about the GALR3 in the human brain, so we were very disappointed that now that they finally get the Galanin antagonist is against the GALR3 receptor. We wanted to have a GALR1 receptor. Of course, now we think it's good that they developed a GALR3 antagonist. And I have, of course, asked if they have tried this in human depression or treat patients. This company was bought by Lundbeck, which is a big Danish company, very interested in depression. So I thought they bought it because of this antagonist. But to be honest, I don't know if they ever tested it in the clinic, in spite of the fact that I've sent our papers to this company and said, why don't you try it? So we will see. So how we think that this could act, I show this now on a little cartoon here. We have the noradrenergic cell bodies, we have the GALR3 receptors, we have galanin in the cell bodies that can be released. Maybe there are some nerve endings also, but in any case, here we have the opening of the potassium channel, noradrenaline release in the forebrain is decreased, and excitability goes down, and then if you have a GALR3 antagonist here, then you would close the potassium channel, noradrenaline in the forebrain would increase and would be an antidepressant effect. That's, of course, exactly what these reuptake inhibitors do. They increase the levels of serotonin and noradrenaline in the forebrain. Now, I think what I want to say with this slide is that to work with antagonists to peptides is probably good, because you can a priori expect that they have fewer side effects. Why? Because we have this idea that peptides are only released if the systems are active. And maybe in the brain you have 20, 25 Galanin systems. And if the disease is selectively active on the locus coeruleus system, it's only this system that will release the peptide Galanin. And it's only at this system that the Galanin antagonist will have an effect. All other Galanin systems are quiet, they are not releasing Galanin, so there are no side effects or fewer side effects. So therefore we believe that peptide antagonists are good drugs because they probably have less side effects because they only act on activated systems that release the peptide. So is there any other support for Galanin involvement in depression? Yes, there is. And this is the result of a big European study which we called New Mood, which was guided by Bill Deakins. And we were 14 groups, and they decided to make a candidate gene study. That means it's not a GWAS study. So you don't look at all, you look at selected genes. And I said we have to look at Galanin and the Galanin receptors because we are so interested in that. So that they did. And this was a big study in Hungary and in Manchester in UK. And they looked at, they made interviews with more than 1,000 patients, and they asked about their history, what they have been exposed to, and so on and so on. And then the result came out that actually they found an association between Galanin, Galanin receptors, and depression, anxiety. And so variants in genes for Galanin and its receptors, all three actually confer increased risk of depression, anxiety in people who have experienced childhood adversity or recent negative life events. So here is a very clear association between Galanin stress and mood disorders. So one more actually evidence for involvement of Galanin. So here I just listed, and you don't have to read it, the milestones which, how this has developed. Many of the drugs that are used in psychiatry, they are serendipity, the result of serendipity. But here's, in any case, a kind of a logic development from animal experiments to studies on the human brain and so on. And of course we don't know if there will be a drug, but that's of course my hope. We'll see. So I think I skipped this. I talked too long time in any case. So the conclusion is that if you look at the noradrenaline systems, we have Galanin synthesis in both rats, mice, and humans. And so it's a conserved coexistence situation. But when it comes to the receptors, there are distinct species differences, and they may be important for identification of targets for drug development. And the Galanin may be regulated at the genetic and transcription level in depressed, suicide, anxious subjects. And especially the GALA3 receptor is a potential target for antidepressants. So here we have an example of a system where a coexisting peptide has a function to protect against too much stress. It's a part of a resilience mechanism, but when this goes wrong, when it is not enough, then overexcitation can cause maladaptation and development of the disorders. So, a long story. Signify we knew in the beginning very little about neuropeptides, and I have many times been running out of grant money, but we know a little more, and there are actually now some drugs on the market which are acting via peptide receptors. This the substance P antagonists that failed for developing for treating depression is now used actually for treatment of emesis, for chemotherapy-induced emesis. It's actually quite efficient, and it's an important application. So in any case, Merck could use it for something, even if it failed for the big disease for depression. I don't know if you have heard about orexin. I mentioned that peptide, which was discovered in 1998, and now, just less than 20 years after, there is a drug on the market for treatment of insomnia, because I think maybe you know orexin is important for sleep control. It's something that is missing in narcolepsy, the sleep disease, and now there is so that is one example of successful treatment of a peptide antagonist. We now know that antibodies to calcitonin gene-related peptide, which is a peptide present in the pain neurons, is very close to approval by FDA for treatment of migraine. And there is also NK3 antagonist, substance P3 antagonist, that may be for treatment of hot flushes. So whether Galanin antagonists will be ever used for treatment of depression, we don't know, of course. But in any case, we have come a little closer to the discovery of the wheel. So I have, of course, had many collaborators over the years. And this is maybe 10 years ago. I had a bigger group before that. I have had support from many sources. I've collaborated with many groups outside my own group. And now the group is much smaller. And we are only 3 now. And I'm fearing that soon it will look like this, you know. Only me. Thank you very much for listening. That's it.
Thank you, Professor. Now we have a Q&A section. Any question? Yes, please.
It's a wonderful talk. So, in the local cellulose, you have neuron that have Galanin, and they also have Galanin receptors. So those receptors, you see the Galanin projection from itself or from somewhere else?
Yeah, we believe that in the rat we know that it's a R1 and the R2 receptor. And if we do electrophysiology, we apply a GALAR1 agonist, then we get this effect. And Galanin by itself also, this inhibition by opening potassium channel. If we apply a GALR2 agonist, nothing happens. So even if we know that the mRNA for GALR2 is present in the locus coeruleus, so we believe that that's a presynaptic receptor that is transported out into the nerve endings in the prefrontal cortex and other parts of the brain. The problem is that no one has so far, as we think, produced an antibody to a specific antibody to any of the Galanin receptors. So we have not been able to study the localization of the receptor protein, only always the transcript. And that's, of course, a big disadvantage, because we cannot show where the receptor protein is. And we have tried to make antibodies. I have bought probably, I paid so much money for commercial antibodies that claim they are GALR3 or GALR1 or GALR2 antibodies. But when we compare with the distribution of the mRNA, it doesn't fit. So I mean, we have very nice antibodies to substance P receptors, to NPY receptors and so on, but for Galanin it hasn't worked. And I think for those who are in the field know that there have been written papers about that. Virtually 90% of all papers published with antibodies to these 7 transmembrane GPCR receptors are wrong, quite simply, because it's very difficult to make specific antibodies. That's sad and bad.
Any other question? Yes.
So there are also a lot of Galanin neurons in the prefrontal cortex. So we know that the locus ceruleus neurons project everywhere in the brain, right? So I don't know anything about the Galanin containing neurons in the prefrontal cortex. So where do they project, and what kind of things do they do?
That's an excellent question. That's a great question. You know, in the rat we have said, okay, maybe there are some Galanin neurons in cortex when we do colchicine treatment and all these things. But what our data here show is that there is a lot of Galanin mRNA and GALAR1 mRNA in the prefrontal cortex. But we have not been able to do localization studies. First of all, we don't have the antibodies. Secondly, we haven't done the in situ hybridization. I don't know if you thought about it, but in this slide where we had the three tyrosine hydroxylase, Galanin and GALAR3, I had shown the exposure times. So it took to show with the situ hybridization maybe a couple of days to show a tyrosine hydroxylase because there's so much enzyme and so much mRNA. But to see the GALAR3 receptors, we had to expose for 8 weeks. So this means the levels are very low. And of course, one problem is that it's post-mortem tissue. And actually, we got brains that had from 2 hours to 24 hours post-mortem times. And we saw the receptor only in brains with post-mortem times from 2 and 4 hours. And you know, it's very difficult to get short post-mortem times for brain. So we plan to do, we think maybe we can show Galanin and GALAR1, because GALAR1 in prefrontal cortex is very high too. If we get some very short post-mortem time. Because we don't know, you know, is it pyramidal neurons where the receptor is? Is it glial cells? Everything is possible. So this is a critical question, but we have got more encouraged, and we believe that the Galanin system is more important in the human cortex than in the rat cortex, because the levels of mRNA are pretty high, both for Galanin and GALAR1 in any case. But we don't know how to. That's why I focus on the locus ceruleus, because there we know we have done the in situ hybridization, which we haven't done in the forebrain.
I don't know too much about depression. I wonder whether there are different types of depressions. For example, some are induced by early childhood trauma, and some like more ongoing stress induced. And whether do you think Galanin is the peptide, neuropeptide that responsible for all the depression, or a particular type of depression?
Also a very good question. And as I said, I'm not a clinician, you know. But I have read and I have understood from my colleagues that actually depression is like every other disease. It's very multi-disease, you know. And I think that this study, genetic study, was a very complicated study, which the statistics was very complicated. But what my Hungarian colleagues tell me is that the Galanin system and association with depression was only in those cases where they had a stress component. So Galanin is not a general, it's not what I'm saying here is not for all depression, types of depression. So it's when stress has to be involved. But then, of course, no one knows exactly what is the percentage of depression that is dependent on stress. But so it's a very relevant question, yeah. Mm-hmm.
In your experiment, you showed that the Galanin expression is upregulated in depression. So I would like to know, do you know the medical history of those the patients? No, I mean the deceased people? Because I ask this because maybe they are taking some drugs that would affect the expression of the galanin when they are alive. So I would like to know, do you know the medical history of them?
Yeah, I mean, this is of course our colleagues in Montreal who have the brain bank. They have all the clinical history of these patients. And we have tried with statistics and so on and they know about the drug treatment. All of them have, of course, been treated with drugs, you know. So, but they at least they came to the conclusion, or we have come to the conclusion that it didn't influence, it didn't depend on the drugs did not play a role in any case. But I think, you see, when you do these type of experiments, you have postmortem times between pretty short days, say 6 hours, but up to 100 hours. Because to get the numbers that you have here, you cannot just take shorter than 12 hours or something like that. So you cannot select, and you cannot select those which have taken just some drugs, and you have to take everything to get the numbers. But so I think this is an uncertain factor of uncertainty, what to say. But
also,
in any case, we try to control for that, and yeah.
Okay, in your presentation, you give me an impression that the Galanin system may be the cause of the depression disorder. But in your experiment, it's just a postmortem brain. So can you propose any experimental design that can show this causal relationship, or is it the disadvantage of the postmortem experiment?
I mean, you see, I mean, so what this is a question of, you have done experiments on rats where you can do things. You have an experiment model, you can test effects of drugs and so on. And so you build kind of the hypothesis of animal experiments. And then what we see then in the human brain is that many things are similar, but there's a receptor that is different. And then we draw all these conclusions. But I think you're maybe I should be more cautious how I present things. But you want to make a case, and so you exaggerate a bit and so on. But one should be humble, and yeah.
Professor, I have a question for the GAL3 receptor antagonist. I'm presuming that the company has it patented, that it's made it not publicly available, right, for the research?
Yes, it is. Yes, you can get it. Yeah.
You can get it.
Yeah, yeah.
So why isn't there like a proposal for the preclinical study where you would use this GAL3 receptor to target patients with depression, since it's publicly available?
Well, I mean, first of all, there's a patent, and the drug company, you see, I mean, this is one reason why there has been so much disappointment in the peptide field in general. So many antagonists have been developed, and of course, the most, what people felt from the beginning was that a CRH antagonist, CRH is the corticotropin releasing factor, and that is the stress hormone, so to say. And everyone thought that if you have an antagonist to that, that would help to treat depression. And many companies have antagonists, and I don't know, many companies have it, and you hear the rumors, oh, now we have it, and so on. But then everyone fails, and most of these small drugs, small molecules, they are liver toxic. And that has killed so many, I think all of these CR1, the CRH1 receptor antagonists have not reached the clinic because of that. And we don't know if this Galanin, there are two types of GALR3 antagonists that they developed. Maybe they are liver toxic. Maybe that's why the companies, they know that now, and they have. And the drug companies, they don't like to tell about failures, because they don't want to give other companies an advantage, you know. So we have done the experiments, we have failed, we have put a lot of money into this. So if they want to try, they should put in the money, so they lose the money. So it's a competition all the time. And therefore we don't know about the GALA3 antagonist. But
Okay, maybe another sub-question is, why is it so difficult compared to the other receptors to make antibodies for the labeling?
I don't know. But also the cholinergic receptors, the antibodies for them, you know, there are papers on that. How many papers in the literature are wrong because they are and the thing is that they don't show, if they didn't show any staining at all, fine, you know, but they show staining and it's false positives. That's the terrible thing. We have a big paper on NPY1 receptors. It's 100 pages in neuroscience, and I got maybe 12 different NPY receptor 1 antibodies, and the first one we did ourselves, and we saw 3 systems in the brain, not very much, you know. And then I got from the United States one that showed, you know, the whole brain was full of receptors, and that was the only antibody. And my student, she worked for 2 years to map this, and still I didn't know if it was specific or not. And then finally I got hold of a NPY knockout mouse. And the day when we put on this antibody in the knockout mouse, I was nervous, you know. 2 years just lost if the staining was there. And we had one antibody that showed beautiful staining, not exactly the same as the one which was good, and that didn't go away in the knockout mouse. And I would probably have published that if I only had had that antibody. So you have to actually have the knockout mouse. Now everyone knows this. But in 10 years ago, people, there were no knockouts, or they didn't care about it. You know, I have published wrong results, so it's...
Thank you.
Thanks for your nice talk. I have a question that, is there any public data shows the exactly 3D structure of the Galanin 3 receptor? Because if we know the exactly receptor structure, we can design specific drugs that may help for the depression.
Yeah. Well, I think drug companies have used this 3-dimensional structure, but as far as I know, not so many drugs have come out from that actually. But I don't know about the GALA1 receptor has been modeled, you know, in 3 dimensions, but I don't know if the GALA3 receptor has. You know.
Thank you.
Very interesting. Thank you for sharing interesting knowledge. I have one question that in your lecture that you mentioned you use antibody to identify the neuropeptide and related receptors. I just wondered whether there is other techniques that can be used to identify the receptor, because I think that actually every part in brain, they are not isolated islands. There are more or less exist hidden collection between every part. So we'd like to give some more suggestion about other techniques can be used to identify the receptor.
Well, I mean, what we did in the early days was to do autoradiography and labeled Galanin with iodine. And then you see the receptor protein. But the resolution of this technique is not so good. So, I mean, so you can see that there is, and we actually did that in the human brain. We saw that cortex contrasting the rat brain is full of binding sites for Galanin. but we couldn't say which type of neurons it is, and we at that time didn't have any possibility to distinguish between the different receptors, because at the beginning we didn't even know that there were 3 receptors before they were cloned. So now we have ligands, so you could do, you could, for example, do the binding and with iodine and galanin iodine and iodinate galanin, and then you could add the GALA2 agonist, for example, to block the GALA2 receptors, and then you would say, okay, this is GALA1, but still the resolution is not good enough to answer the question that I want to know. You know, is it, which cells are it, and is it nerve endings, or is it kind of a diffuse things? But as far as I know, this is the only way to do it, actually. Mm-hmm.
Okay, thank you.
But you should come up with something.
Yeah, Professor King here.
I have just general question about the neurotransmitter, because Galanin is related with, involved in the depression, and CCK is involved in the memory formation. But I'm always curious, the strength of the stress, it decides the synthesis of the neurotransmitter and release. The amount of neurotransmitter is released. In other words, if someone is under the suddenly under the huge stress, it's like they just get depression very quickly, or they have some problem with memory formation. So the strength of the stress, it decides the synthesis of the neurotransmitter and amount of neurotransmitter released. So if neurotransmitter is released a lot suddenly, it have the bigger effect, and then people quickly go to the depression, or it's like neurotransmitter is less synthesized and then less released, people can manage the depression. I'm just curious about the strength of it.
Well, I mean, what I'm trying to say is that, of course, we don't know. We believe that galanin is released also in the forebrain from the locus. If we now concentrate just on the locus coeruleus neurons, it's released in the forebrain. What it does there, we don't know. You know. We know in the human brain there are GALA1 receptors which are inhibitory, but we don't know what neurons they are on and so on. And so but what I'm trying to think now is that the purpose of galanin is actually to be a resilience mechanism, to resist against stress. And that is because we know that the locus coeruleus neurons are activated by stress, that's for sure, you know. And so if they are not activated so much, it's only noradrenaline that is released in the forebrain. And we know that, of course, these neurons are very important for vigilance, for many things. They have so many functions, you know. But the problem is now when they are too much activated. And that's, I think, is the purpose of galanin, maybe in these neurons. That is to keep this activation under control, so they don't run crazy. And that is occurring in the locus coeruleus, to inhibit the neurons. And as long as this inhibition is moderate, so to say, just keep them in a certain range, so to say, then everything is fine. But it's when this is a dysfunction, when it gets too much, when this inhibition of galanin gets too strong, so that they really silence the noradrenaline neurons, then there's no noradrenaline release in the forebrain. And that's the situation when depression can develop. And this is what I wanted to say here. I had this, that I skipped to slide, pain, you know, because there is something similar there. Galanin, as I showed you in the beginning, is upregulated when you have nerve injury, okay? And our conclusion from many studies together with Susanna Wiesenfeld-Hallin is that one important thing for galanin in the pain neurons is to inhibit pain, okay? In the beginning, there's two effects of galanin. In the beginning, it stimulates pain, increases pain. And that's what pain is good for. It's a warning. But if it's too much, then galanin is upregulated, and then it inhibits pain. And that's, of course, what we want. We want pain to just warn us and then stop. But if this mechanism, just like as I say here, goes wrong, if this inhibition is not enough, then we are not protected against pain. And then chronic pain develops. Neuropathic pain that, some people the whole life will have pain. So, I'm not saying that galanin is the only mechanism, but it's one mechanism that is endogenous to these pain neurons, that they have first galanin warns, then it stops pain, but at one point it is not enough, and then chronic pain develops. So again, galanin has this, it's an accessory mechanism that helps, so to say, to control pain, but if it's not enough, then chronic pain develops. And here, this is also something goes wrong. There is a dysregulation, dysfunction, then galanin doesn't help any longer. Or in depression, it actually makes it worse. It develops depression because of that, of the galanin. But normally it's a protective mechanism.
Yes, okay. Professor Hökfelt, welcome back to City University, and thanks for the wonderful talk. I guess we can say that depression is putatively, reward system based. And so I wondered if you could maybe touch on how the Galanin system may interact with the reward system. For example, we talked about the locus coeruleus, but how does this interact maybe with the ventral tegmental area or the nucleus accumbens?
Mm-hmm. Now that's also a very good question, which we never have studied, but other groups have studied that. And they think that there is a projection from the locus coeruleus to the dopamine system of the A10, the A10 group, and that there is this connection, and so they have done some studies, but then you have the connection to the dopamine system and the reward system actually also. So, but it's not. That group has given up.
Based on your experience and work with the peptides, do you think that perhaps galanin solely works through the kind of the locus coeruleus and norepinephrine system, therefore working in the forebrain, or do you think it might also modulate some of the other neurotransmitters? Therefore, it has a dual action.
It could modulate the dopamine system. The thing is, we haven't looked in the human brain. So Eric Nestler is of course one who has championed the dopamine system and depression, but he has never touched galanin. He never mentions galanin, and there is one group in the United States, Jay Weiss, who has actually studied that, and he had one paper. His last paper on that was also looking at the galanin receptors in various systems, so, but it was difficult to draw a conclusion how this was actually. But he had this idea that there is an inhibition of the dopamine system by galanin, but no one has really looked at the situation in the dopamine system. We have never seen in the rat any galanin receptors in the dopamine neurons, actually. So, but then you would like to do this in the human brain to really see if, but it's a very relevant question.
Over there, it's young
Sorry, hi Thomas, nice talk. And I have two questions. First one is, do you think there are any other upstream pathway that control the galanin system in healthy? And because I think if in healthy people there should be a regulation on this galanin system, but in depression, this patient, probably this control system is not functioning very well, so the Galanin system just overreactive, and so that's why I wonder whether there is an upstream pathway that control the LC Galanin system. And the second question is there any difference of mechanism on the release of Galanin from terminal and from the soma? Because usually we believe that the neurotransmitter and the modulator are usually secret from the terminals. So this is the first time that I know that neurotransmitter or neuromodulator can be released from the soma. Yeah, it indicates that this Galanin release from soma can activate the terminals from other parts. I mean, nearby the soma, Galanin positive neurons. So this is another question. Thanks.
Yeah, I mean, the evidence for dendritic or somatic release of Galanin in the locus coeruleus is not very strong. I mean, we, as I said, we have a paper on that, but compared, for example, to the situation in the vasopressin oxytocin systems in the hypothalamus, the evidence is not. They have fantastic, nice studies showing that the soma of in the paraventricular nucleus is releasing vasopressin and oxytocin, and actually there is a local control of these neurons which is completely independent of the situation in the posterior pituitary where the nerve endings are present. You know, so that's a strong evidence. There is actually a very nice paper on the release of noradrenaline from the soma of the locus coeruleus with you know, real high-tech technique showing a single vesicle release, but this cannot be done with Galanin. So the case for that is of course not so strong. So I don't know if you also then asked if Galanin released from the soma could act on other nerve endings and so on in the locus coeruleus region, or if it's only on the autoreceptor, so to say, that is produced by the locus neurons themselves. I don't know if there's any other target, so to say, for somatic dendritic release of Galanin there. And then what about the, I mean, the locus coeruleus is controlled by so many things. I mean, for example, pain. It's, you know, if you distend the urinary bladder, locus coeruleus activity goes up, and so it's so it's not the if you mean upstreams. I mean, this is now downstreams from the sensory input to the locus coeruleus. If you mean if there is activation from descending systems to the locus coeruleus, there of course are descending systems, but how they are involved in, you know, if you look upon the innervation of the locus coeruleus, there is a lot of various types of transmitters that innervate the locus coeruleus. One is the CRH neurons, the cortical releasing hormone, and they are activating the locus coeruleus neurons. So that would be a connection between the CEST system and the locus coeruleus. Another system that is really important for the locus coeruleus is the orexin system that I mentioned, which is activating and that's how sleep wakeness is controlled, part of that control. So locus coeruleus is of course also very important for sleep and wakeness, and so you know, it's a very, very system that I mean, when we in the early when Kjell Fuchse discovered that locus coeruleus has noradrenaline, and when we saw that it's innervating the whole brain, which no one believed in actually, because the classical, in that time, in the 60s, the classical neuroanatomy said there is no system that ascends all the way from the lower brainstem to cortex. There are no such systems because they couldn't detect them with the silver techniques that they used for tracing. So when Shell went to congresses and said that, he got enemy with many of them because he knew this was correct, because he could show that there is a pathway like this one. And then we started to find, how can it be that one system can release noradrenaline all over the brain and has some sort of specificity? And at that time we didn't know about different receptors. So of course it's the many types of adrenoceptors that then, and in which cells they are cause the selectivity, so to say. But still it's such a complex system, and I give such a simplified picture of it, so it's a bit embarrassing. But I really like all these questions, because normally when I give talks, no one asks a single question. So thank you for
the last question.
Interesting.
Sorry, it's late, but I thought this is an important question for students, because we have the first scientists discovered the co-release of neurotransmitters and neuropeptide, single terminal. Now in the locus cellulars, we know that the neuron release norepinephrine, they release galanin, and recently the paper saying they also release dopamine. So they possibly release three neurotransmitter and neuromodulators. Can you give us any suggestion how they're controlled? Their release is controlled.
But as you say, that was also very surprising, that they released dopamine.
Yeah. Maybe you can discuss over the lunch. So,
to be honest, I have never said that I discovered co-release, because we never studied release. I only did one thing. I saw that they are present in the same neurons. And since there are vesicles, we always assumed that they are released, so. But
let's give a warm applause again. From IAS to Professor. Oh, dear. So want to take a picture? Thank you. Let's come to Professor.